next and last presentation again from Dr. Neil Desai. The title of this presentation now is New Clinical Directions with AB009, a targeted nanoparticle mTOR inhibitor. I won't read his function in which he presents this. I think he will explain to us. Go ahead, please.
Hello, the mic on. Okay. Well, thanks again, and it's a pleasure to present to you for the second time this morning. Here we're going to talk about, you're all familiar with the Abraxane, so this will not be a stretch to explain. What we've done is taken an mTOR inhibitor, which is rapamycin or sirolimus, and then using the same technology, trying to develop that in the clinic. And this is in a different company. It's not Celgene, it's AADi, which is its own independent company, and we've licensed the molecule out of Celgene into AADi. So what are we doing? We're building on prior success. Abraxane's done well in the clinic and on the market, and the platform is validated in terms of albumin-based delivery of nanoparticles. And so we're using different molecules to try and see how we can gain some advantage over existing technology or existing drugs. This is sirolimus or mTOR inhibitor, well known, previously approved in the transplant setting. You know about other mTOR inhibitors that have been approved in cancer. So again, the biology, et cetera, is very well described. We're finding new ways to apply this and based on novel properties of the nanoparticle. The difference here is what I want to do in this company is focus mainly on rare diseases, which have not been sort of well taken care of in terms of approved therapies. So mTOR pathway, again, I don't really need to go into the detail, but the important aspect of it is sort of right here at the bottom. The mTOR inhibitors have many sort of pleiotropic functions. You're, of course, familiar in the transplant setting of immune suppression and in the cancer setting, but there's metabolic immune modulation and other functions that can be leveraged to develop new indications. So this sort of gives you a picture of where we can apply these agents. And some of the functions of mTOR inhibition, what it results in, of course, is decrease in mRNA translation. It affects the cell cycle and decreases proliferation. And as a result of that, you can find certain cancer indications where you can apply this. And we're both in adult and pediatric cancers. But even outside of the cancer space, there's some very interesting indications you could go after. For example, pulmonary hypertension. So I'll talk to you briefly about that today. In the immune modulation space, clearly we know rapamycin as an immune suppressant, but there's other aspects to its immune modulation, which is immunostimulatory effect of CD8 positive T cells and increase in T cell memory. So we can leverage that, which has not really been done currently in the clinic, and see how we can use that opportunity to combine with immuno-oncology agents, cancer vaccines, and look at immune dysregulation. And then there's a whole very interesting space of the metabolic aspect of the mTOR pathway that we can leverage, and that gives us potential use in mitochondrial diseases and even in aging. So for today, I won't be able to discuss, of course, all the applications, but I'll focus in on a couple. And this is just prior data that we published. We finished a phase 1 trial, and this was published in Clinical Cancer Research. And it was 26 patients. We established the maximum tolerated dose at 100 per meter square given every week, which is much higher than the conventional mTOR inhibitors. We saw the evidence of activity in these patients. We had 2 RCC patients which had very nice tumor shrinkage, and then we had very long stable disease in a series of different cancers, neuroendocrine cancer, mesothelioma, and bladder cancer. And importantly, the safety signal was significantly different from what we are conventionally used to with mTOR inhibitors. We also saw the target inhibition that was measured. So let's see what the difference is in the PK. So here's a comparison. This is our phase 1 PK at one of our doses, which is 45 per meter square per week. And compare this to published data on oral rapamycin, for example. So there's clearly a huge difference in terms of the peak levels and the overall AUC. But the important thing to note is that oral mTOR agents are susceptible to very poor absorption, highly variable absorption, with diet, without diet, and as a result, you need therapeutic monitoring. So you have to maintain a certain blood level, which is typically what they try to do is between 5 to 15 nanogram per mil. You have to titrate the patient on a weekly basis. So it becomes difficult. And in addition, there's a lot of toxicity baggage that goes with that, heavy GI toxicity, for example, which causes patients to come off drug. So we've discussed this mechanism in the past. This is the albumin-based mechanism that allows the drug to get through the endothelium and into the tumor cell, in this case, through the caveolar pathways. And we published this information for Abraxane in the past. So we can look at a comparison of activity versus oral rapamycin or oral everolimus. This is an aggressive bladder cancer model, and the dosing was designed so that we could pick out, with the aggressive tumor model, we can differentiate between different drugs. We treated, you see the first 3 here, they're all at the same weekly dose, a total of 15 milligram per kilo per week, but yet you can see the clear separation between ABI 009, which is the NAB form of rapamycin, versus the other mTOR agents. And similarly, you can see that in survival. We've also previously presented across a variety of different tumor models, whether it's pancreatic cancer, breast cancer, colon, and multiple myeloma, that ABI-009 is quite active as a single agent. And then when we can combine with multiple different agent types, like HDAC inhibitors, EGFR, AKT, or even anthracycline like doxorubicin, to get nice combination effects in these tumors. So the question is, where do we go from here? So we have started working in the clinic in different indications, which I'll talk to you about. And so our lead indication is what's called PEComa. Many of you may not have heard of this, but this is a very rare form of sarcoma, and I'll talk to you about this indication in a little more detail. In pulmonary hypertension, and again, this is a relatively rare indication. It's an orphan indication that needs additional therapies. In rare pediatric tumors, there's, you've heard of the Ewing sarcoma, rhabdomyosarcoma, medulloblastoma, etc., where mTOR is an important target to suppress. And so we plan to do combination studies here with existing therapies. And then there's several other oncology indications that we're looking at. We're looking at hematologic indications and immuno-oncology combinations. We also have a grant from the National Institute of Health, National Cancer Institute, for early stage bladder cancer, where we are putting the drug directly into the bladder. So in the interest of time, I'm going to discuss just 2 indications with you. PEComa is the first one. Sorry, there's a lot of language on this slide, but basically it's a very rare form of sarcoma. And the characteristic and the diagnostic tool that's used to identify this type of tumor is that it has both melanocytic characteristic as well as smooth muscle cell characteristics. So that's the diagnostic test with these antibodies that you use to identify a PEComa. And it's a relatively recently defined entity. In fact, just in the last 10 years, there's been a WHO guidance to identify what is a PEComa. And in malignant PEComas typically involve the lung, as well as the gynecologic and GI systems. And the limited data that we have we can say that the estimated survival once you're diagnosed with metastatic or advanced disease is about 12 to 16 months. So it's like, there's a limited lifespan overall survival issue. There's not a single approved drug in this space. And mostly it's treated like any other sarcoma where doxorubicin or doxorubicin-based regimens are used, and with a 10 to 15% survival, as Felix just mentioned. The interesting thing is it's a very rare disease. There's only about 200 to 400 patients maybe in the US, similar in other regions of the world. And so we're going after this extremely rare disease where there's no prior approval. So we went to the FDA, and I'll explain to you the rationale behind why we would want to use mTOR inhibitors here. But we confirmed with the FDA that we could do a single phase 2, a single-arm trial, open label with 30 patients with a response rate endpoint for approval. And so we've got that under our belt, and we're enrolling in that trial. So just before I get into some details of that, here's the pipeline that we're looking at. So advanced PEComa, we're in the registration study with a small phase 2 study. We have a solid tumor program that's going with the Mayo Clinic that's just initiated phase 2. We have a bladder cancer study sponsored by the NIH that we're finished the phase 1, and when we're starting phase 2 as a combination. We're working with the Children's Oncology Group to initiate a phase 1 and a phase 1b study in pediatric tumors and hematological cancers also, and pulmonary hypertension. So quickly, mTOR pathway, in majority of the PEComa, the TSC2 are mutated, and therefore results in activation of mTOR. So it makes very good sense to go in with an mTOR inhibitor to shut down these tumors. And this is the phase 2 registration study. Our endpoints, really our primary endpoint is objective response rate. And then we plan to enroll 30 patients. We've initiated the trial just in the last couple of months. So the next indication I want to discuss quickly is pulmonary hypertension. It's again a rare disease. It's based on proliferative biology inside the blood vessels of the lung. You see this proliferation in patients with pulmonary hypertension. So the survival of these patients, depending on which functional class they are, especially in the functional 3 and 4, is quite poor. So functional class 4 has a median survival of about a year and a half. So this is like cancer, in fact, but inside the blood vessels. And this is what the biology looks like. So this is, you get proliferation inside. This is a normal blood vessel. You get proliferation, you get remodeling, you get thrombus formation, fibrosis, etc. And we can treat this. The current treatments, there's several approved drugs, but all of them focus on vasodilation. So how do you vasodilate? So the nitric oxide pathway, the ETRA pathway, to vasodilate these blood vessels to get symptomatic relief. But that doesn't treat the root cause of the problem, which is the proliferative and fibrotic aspect. So when we come in with ABI-009, which looks at all of these aspects, we can add this drug on top of the existing therapies and treat the disease. And we have an animal model that looks at some of this. So in an analogous disease, which is restenosis, you're familiar with. So these are arteries in pigs, and these is a pig model of injury where you get proliferation inside the blood vessel. So the control animals which receive no treatment have extensive proliferation. You can see that when we treat with nabrapamycin, this is greatly reduced. So we know that the proliferation can be stopped with the right drug inside these arteries. So we're about to initiate a phase 1 trial in this disease. The primary endpoint is, of course, safety of 16 weeks of ABI-009, and the secondary endpoints are, critical endpoints for pulmonary hypertension, like peripheral vascular resistance, etc. And we will include the class 3 and 4 patients. So we expect to enroll by late summer or fall. So I will stop with that, and I have the acknowledgments, and this is a view in Pacific Palisades, California, which is close to Malibu and Santa Monica. I'm sure all of you are familiar with that. Thank you.
So thank you very much for this further presentation. Are there questions for this presentation? Yes, please.
Yeah, interesting talk. And why did you choose in the very orphan disease, very small market?
Because there's nobody to go after them, right? Somebody needs to help these patients, right? And the large companies typically go after the big markets, the lung cancer, the breast cancer. So somebody has to look out for the the people who don't have anything. So that's my motivation.
Maybe I can catch on with that just before. You mentioned also restenosis. This is not something which is on your radar?
Yeah, it is a good question. It is on our radar, but I mean, again, those are big markets. So I'm trying to focus the company on sort of smaller markets. There's an added complication there of you have a device-drug combination. So of course that can be overcome, but right now I think the focus of the company is sort of on the orphan diseases.
Okay, thank you.
Neil, I have two questions. The first one is regarding the immunological effect of mTOR inhibitors. We tend to recognize them as immunosuppressive agents, and we use them actually in the prevention of graft rejection in kidney transplant patients. So I couldn't really put together this. I couldn't reconcile with one of your slides in which you show as if there was an immune enhancing effect, immunoboosting effect
Yes.
of your compound. This is one thing, and the other one has to do with the pharmacokinetics. You're actually giving once a week a high dose, and patients usually get a daily dose orally. So what's going to happen? I mean, are they going to be underexposed? They will have a big peak exposure and then be underexposed for several days until you give the next injection. So it's like completely different drug. Can you comment on that?
Yeah, sure. So you're right about on the immune aspect. What's traditionally known or recognized is in the transplant setting that you can prevent graft rejection. But if you look at the data, it's very interesting. The rapamycin by itself is not a very good immune suppressant, actually. It's usually used in combination with cyclosporin in kidney graft rejection or prevention. But there's a whole other aspect of rapamycin and mTOR inhibition that results in stimulation of T cells. There's increase in autophagy, which is a very important aspect, that rapamycin inhibition or mTOR inhibition drives autophagy, which, if you know about autophagy, it helps antigen presentation. There's data in the literature also where if you use it in concert with a vaccine, it acts as a very strong adjuvant, and it's shown that you have memory T cells that are activated to help in that setting. So there's these other pleiotropic effects, immune effects, that have not been really utilized that we can leverage. And there's some very nice papers that show that. So I'd encourage you to look at that. On the PK aspects, the profile is completely different from oral, as you know. Oral, you just get a basal level, and then you have to try and maintain that over time by daily dosing. So the PK aspect is, we've done extensive PK in the phase 1 study, and we don't get below the subtherapeutic levels. So you get the peak level, the half-life is about 60 to 65 hours, so you maintain it over an entire week, and you can adjust the initial dosing to maintain and stay in the therapeutic range. And by the time day 8 comes along, then you redose, and then you get the peak. And then we think that having that peak actually helps drive drug into the tumor. In addition to that, we have sort of the caveolar transport mechanism, which is not there for just standard rapamycin or everolimus. So, and we showed in the tumor models that when you compare the two, that at the equal same dose on a weekly basis or equivalent of a weekly basis, that you get a better tumor suppression.
Do we have more questions? Yes, there's one more.
Do you hear me? Yeah. So maybe following on this first question, I think that there is a very interesting link that is also related to the properties of the mTOR to switch the metabolism from glycolysis to the oxidative phosphorylation, which is actually a property that is mostly taken by regulatory cells and maybe Th2, but not by Th1 and Th17 cells. So maybe this has to do also that some memory cells could be spared, but not the ones that they are maybe the more inflammatory or the ones that they will promote the responses like in the graft versus host or maybe even in the tumor response. So I will say this is a borderline that has to be considered. I don't want to say it's impossible, but this is one of these that the balance is very delicate.
No, it's true, but there's a very nice paper in that's published on exactly that, where they're looking at the graft versus host relative to sort of the adjuvant effect of a vaccine. They've done it in the same study. So it shows that it doesn't affect the graft versus host, but you get a big stimulation as an adjuvant effect. So I would encourage you to look at that paper, and it'll answer your question.
Okay. If there are no more questions, I think we're at the end of this session. I would like to thank all speakers of this early morning session again for the contributions and also the audience for the participation in the discussion.